Okay, everybody, welcome to Bat ID Part 2, the Advanced Bat ID. I am going to start sharing my screen. Dun, 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 dun. Yes, all right. Okay. Bear in mind that I can't see the chat, so Becky's going to monitor the chat and Scott is going to annotate the photos. What I'm going to do is like a 1 minute whiz through, just a refresher of what we did last time. Just to kind of jog your memory, because I know it's been a month. So as you know, I've now put all of these videos on the website, password protected. So if you need the login or the link or anything like that, just give me a shout later on. So last month we went through basic Bat ID. Come on, computer. There we go. Okay. So I want to move this up a little bit there. Okay. So we went through the UK species, and we went through our county species, of which there are 12. And we were talking in general, which we were driving home quite a lot throughout the talk about going on anatomy rather than pelage. Although pelage can be a useful indicator of what you're looking at. Basically, because pelage is subjective and it's variable, and it can vary with the age of the bat and the season and the time of the year. And bats go through molt and various other things, so we don't go on pelage. We did a bit of basic anatomy, showing you what the bat's wing bones are in comparison to human bones, and basically that talking about how Chiroptera means hand wing for a very good reason, and that when we're talking about the different digits, proximal is closest to the animal, and distal is furthest away from the animal, with the animal's hands, palms facing towards you like jazz hands. And then we did a bit of basic anatomy. The main things that we were looking at were the calcar, the nose and the nose leaf, if there is one, the tragus and the thumb and the fifth finger when it comes to non-sexual assessment of age. And we were looking at all the different types of tragus shapes, which is your first step in identifying your species, and looking at the different types of calcar and the presence or absence of the postcalcareal lobe, the anatomy of the nose leaf, and basically how to separate each of your common species. So we went into separating lizards and noctules. We went into separating pipistrelles and serotines, which is just massive. And then we went into quite a bit of detail about the wing venation of pipistrelles, and all of the pipistrelle features. Remember that we do these collective. None of them are exhaustive or 100%. So we looked at the wing venation of each of the species. And showed you some illustrations. And then this is a general look of color, but we don't go on pelage, but it is an indication. Comparative wing venation, internarial ridge, general coloration of the genitals being orange in Pygmaeus and cream in Pipistrellus. And then we went into looking at separating Myotis bats, which have a straight tragus, and looking at separating natterers and Daubenton's. Basically, these are large Myotis in the West Midlands. We don't yet get Bechstein's bat, so we didn't include that in this separation. And here you go with your comparative natterers on the left, looking at the curved calcar with golden bristles on the natterers versus the straight calcar and giant hairy feet of the daubs. And then looking at that sort of half moon of pale fur at the base of the recurved ears of the natterer's bat compared to the lack of color, that lack of paleness at the base of the dark non-recurved ears of the Daubenton's bat. And there's the same thing in the hand. Again, natterers on the left, daub on the right. And then we're looking at separating natterers and Bechstein's bats. I went into quite a bit of detail about the length of the tragus, the number of transverse folds, but basically they are more or less the same size and weight. and it all comes down to the ears and the calcar. You won't really get, it's very unlikely you'll get any chance to handle any Bechsteins on our project, so it's just good to know. And then small myotis. This is separating whiskered and Brandt's. Again, we don't get Alcathoe in the county yet, so you're unlikely to have to worry about separating those. So we're just separating them based on penis, tragus, ear color, pelage, but not really pelage, presence or absence of a protocone, and looking at the comparative dentition in the lower and upper premolars, and possibly looking at the forearm length. So just to skip past all of this stuff, wee whiskered, big bulbous Brandt's. So one of the things that I was talking about in a bit more detail to Lee to help her remember which one of these, if I put this in front so you can see me, if you look at the video of me rather than the screen, if you think of whiskered, we're talking about the convex and concave edges, outer edges to the tragus, think of whiskered as being like a W and Brandt's as being like a capital B, like going on its back, to help you remember that in a whiskered bat they're pointing outwards, in a Brandt's bat they're pointing inwards. And then protocone stuff, I would suggest you just watch this bit. I'm going to timestamp all the different bits of the video for you when I get a chance. I'll probably do it at the same time as I do this one. And then the dentition looking at P3. And then these are the sex-specific 95% confidence intervals. With 95% confidence, you can separate whiskered and Brandt's based on the length of the forearm by sex. Not 100% of the time, but. And then looking a bit at other species, Nethusius, Barbastelle, Alcathoe, greater mouse-eared, greater horseshoe, grey long-eared, and vagrants, none of which we get in our county. So that is your basic run through of everything we went through in detail last month. So now we get into the more detailed identification. So we are going to go through the reproductive cycle of Vesper bats. We're going to go through determining sex, which in most cases is pretty much like, whoa, it's a boy or it's a girl. It's very, it's pretty easy. The only time that you would think that you have a male when you've got a female is if you've never seen a male before. It's literally one of those things where they're pretty well endowed for their size. And then we're going to look at determining breeding condition and inferring age class from that breeding condition, and look at non-sexual assessment of age. So we'll also take a little bit of a look at ectoparasites and health assessment. And then most importantly, before we crack on with our survey season, which we're hoping we'll be able to do, because it looks like things might be easing up a little bit in the next month or two. So we're anticipating being able to carry on with our survey season. So we really need you to have good biological recording principles ingrained in your brains. So sex, age, and breeding. This is male versus female. It's either got a willy or it doesn't. And this is about as small as they get, and it's still obvious. So there are, thank you, Scott, that's its willy, yes. Now with females, if you are not used to seeing males, and say your first bat that you handle is a female, they can be distended, especially as just before and just after they give birth. And so it can look like they have a bit of a protuberance there. Also, if they're in poor condition, if they're quite skinny, it can look like they have a bit of a protuberance there. But it is quite obvious once you start seeing males that you can separate them pretty easily.
Or if it's just a really mature female that's had lots of young.
Mm-hmm. Yeah. One of the other things in the species that have them, which is Nyctilis and Pipistrellus, is the buccal glands, which are the glands either side in their mouth. They're basically just fatty tissue. They're not actually glands, but we're doing some work looking into the chemical composition of the volatile chemicals that they have, and they basically are much more prominent in males than in females, and much more prominent during the breeding season. In the case of noctuals, they've been documented anointing their roost entrances using these. So they basically do like what a cat does, when a cat rubs on you and to get its smell on you. So these are very prominent in breeding males of the species that have them. Okay, so a reproductive cycle in temperate Vesper bats, and that sounds very specific, but it is very specific because of those of you who will have watched Scott's talk on the bats of Costa Rica, that he did a bit of talking about how you get polyestrous species, which will breed multiple times in a year, but they don't have to hibernate, so they don't have to worry about it. Whereas our bats in the UK are all very constrained by time, so they have to hibernate for X amount of time. They wake up when conditions are right, and they then have to get their young born, weaned, flying, and fattened up for hibernation before the next hibernation season. So it's all very, very squashed in, and all very very high pressure from the bats point of view. So right now we're in hibernation period. So it's hibernation and sperm storage, and sperm storage is basically because the bat's mating season happens before the hibernation season. The females store the sperm inside themselves, and they use delayed fertilization. So this is not delayed implantation like you get some species like badgers will they mate, the sperm fertilizes the egg, and then they store the egg and don't let the egg develop until it's the right time. In the case of bats, it's delayed fertilization, so they literally just store the sperm. Another thing that goes on this time of year is male bats will wake up and mate with hibernating female bats. And as we go into talking about the epididymis and the testes, it'll become a bit more clear as to what's going on at what times of the year with their testicular swellings. But basically, there'll be some mating going on now. In kind of, depending on the temperatures, kind of April-ish, they emerge after about, we think, 48 hours-ish of being in good conditions with food and warmth. They will become pregnant. And depending on the species, X amount of days later, something like 45 days for Pipistrelles, but it varies by species, the pups are born. And this is why we don't do any trapping between June to mid-July. And if we have a very early spring, we tend to, as soon as basically everybody is watching, say, Facebook and social media, and everyone who does trapping surveys, as soon as someone catches a pup, everyone hits the brakes until mid-July. So that can happen in the end of May. But generally speaking, pups are born in June, and they begin flying within about 4 weeks. So then they spend the rest of their summer before hibernation fattening up. Swarming happens in the autumn. Swarming is something that happens to certain species do and other species don't. So we can go into a bit more detail about that. But basically, swarming happens, we think, for mating reasons. Bats tend to swarm where they hibernate, and also for social reasons, because the bats are very gregarious and they come together, and this swarming is the time of year when they aggregate. So, pardon me, I've got a sore throat today. So basically, particularly your Myotis species and your brown long-eared bats are your swarming species, and they get together in the sites usually where they're going to hibernate. And when it comes to the males, they experience a drop in their weight during this time. Whereas the females, after their lowest point of weight, the females is obviously after they've given birth, and after when they're weaning, they're exhausted, they're feeding a baby, and so they are at their skinniest at the time when their baby is just starting to fly. Whereas with the males, they don't have to worry about weaning and nursing, so they feed throughout the summer, and then they drop in weight in swarming season. And the reason basically is just because at that time of year they care more about sex than food. It's more important to them, because the whole point of eating to stay alive is to facilitate having sex and having offspring, and so that becomes a priority for them. And then they basically, after the swarming season, they have a very short period in which they can fatten up before hibernation. Now, for species that are gleaning bats, I'm not sure whether I put anything in about feeding gills, but basically gleaning bats are bats that will pick insects off of surfaces, as opposed to hawking bats. and open aerial foragers, which take insects on the wing. So you have species like Natterer's bat and brown long-eared bat, which are specialists in gleaning, and so their food is around for a little bit longer than the food of the bats that are taking insects in the air, and so they can push their swarming a little bit later, and so they get their dip in their weight happens a little bit later than the dip in the weight of the bats that are after the flying prey, because basically they have to eat and fatten up before hibernation. So with Natterer's bats and Baileys, they can push it a little bit. Okay, so, oh yeah, sorry. And then after swarming, mating happens, and then we all start again. So this is more going into detail on what's going on with the testes and the epids. So I'm going to talk a bit more about the terminology in a second, but basically in August, the size of their testes will peak. And this is obviously because they are creating all of their sperm then. And as they go through this late summer, spermatogenesis happens, they create the sperm, it all gets, the testes fill up. And then in late summer, the sperm are leaving the testes and enter what we call the epids, which is the epididymis, the chorda epididymis, for storage. And it's for storage so that they can wake up in the middle of the winter and mate with sleeping females. So by sort of hibernation time, the testes have shrunk, but there is still sperm in the epids. So in spring, they wake from hibernation, the epids shrink, we get, and when we are surveying most of the time, depending on what we're doing and depending on COVID restrictions at the moment, because we basically have to absolutely minimize the number of time, the amount of time that we are holding bats for. And so we're only getting absolutely essential information at the moment. So but when we're collecting a full data set, we record the relative size of testes and the relative size of epids as well on a scale of 0, 1, 2. So 0, they're pretty much non-existent. 1, you can see them, and 2, it's like they're very obvious. So in spring the epids are shrinking, and then it all starts again in kind of August time. So Scott, if you can annotate this, that'd be great, because you can see in this picture the testes and the epids, and you could be forgiven for looking at this picture and just seeing those two little balls and thinking those are the testes, and they are not. The chorda epididymis is a tube that connects the testicles to the vas deferens, so that basically they are they're at the base of the tail. So if you find the tail of the bat right up the top near the base of the tail, there is a little tube on either side, and it's either full or it's not full, and they can be very, very hard to see if the bat is not in breeding condition. But they also vary in color. They can be pale, they can be dark, and they can be pale with dark tips. And those three conditions of coloration of the epididymis can tell us how old the bat is with a relative degree of precision in at least whether they are a baby, a 1-year-old, or older than a year. So yeah, you can just about say there's the darkness of at the bottom of these epids. There's a few more pictures that we can show you, but basically, that the testes are the big swollen lumps above, and down either side of the tail are the epids. And then we're going to go into a bit more detail about coloration here. So basically, the idea of the epids, it frees them from having to time their mating with the female being fertile at a good time of year. So basically, they can at their leisure mate during hibernation, but they will also have had a proper mating season. And then the females can they all become pregnant at the same time as soon as conditions improve. So it doesn't matter when they were mated, whether it was in the late summer, whether it was during hibernation, they all still get pregnant in April and have babies in June-ish. Okay, so the reason that the epids change color is because of the breakdown of the tunica vaginalis. So this is basically a sheath over the epids, And during the bat's very, very first breeding season, when that sperm flushes into the epids, it stretches this tunica vaginalis, and it causes the color, the pigmentation in it to break down. So after their second year, the epids will look completely white after their second breeding season. And most of the time, if this is accurate. It's not accurate for some species. I'll go into a bit more detail in a second. But basically, here are some. So the bat, the big picture of the bat is an epid that is epids that are pale with dark tips. You can't see the willy in this picture because the thumb is holding it out of the way, so don't be fooled. It's not a girl. And then top right is the very orange swollen epids of a soprano pipistrelle, and then bottom right are dark epids. So hopefully you can see the difference between pale top right, dark bottom right, and then pale with dark tips, which is basically just the very, very tip of these epids have got a dark patch. So what this tells us is the breeding, the relative age of these bats, with a few exceptions. So when they're born, they have dark epids. A bat with dark epids is definitely a juvenile. A bat with dark epids basically means it has not undergone spermatogenesis, so it is sexually immature. And a bat with PDT epids is a yearling. Now, there are exceptions to this, but generally speaking, if a bat has pale with dark tips, it has gone under spermatogenesis once, which basically means it is the year after the year it was born. However, in a good breeding season, some bats, brown long-eared being one of them, will undergo spermatogenesis when they're 3 months old. So in their first year. So we cannot always say that a bat that's pale with dark tips was definitely born the previous year, because in a good year, healthy bats may happen to them in the first year they're born. And so what we do on our project is, rather than saying it is a juvenile or a yearling, we just say it is sexually mature or sexually immature. We do record whether it's PDT or pale, so that at some point in the future, if we want to differentiate between those, then we'll do that. So we always record pale, dark, or PDT. But if we are doing analysis about whether a bat is its maturity, we go on sexual maturity rather than approximate age. So here you go. So 29% of brown long ears at 3 months can show some kind of epididymal distension. So it's basically the size and shape of epids is better criteria for determining sexual maturity. But then bear in mind as well, we are mostly talking here. We're talking about Vesper bats. The Rhinolophidae is very different. They greater horseshoe can take up to 5 years to become sexually mature. So you can't do this kind of it's a yearling thing with those species. But generally speaking, I mean, we don't catch very many lesser horseshoes, and we don't get any greater horseshoe bats. So it's not really an issue for us, or a comparison that we have to really make. So there it is. So basically, it's a general rule of thumb about determining the age class of these bats. But just remember that, like almost everything, there are exceptions to the rule. Okay, females. So females can mate in their first year. It's not like they have to produce sperm or do anything to get ready. So they can give birth the year after they were born. So we know that a pregnant female is at least 10 months old, but that's all we can really say. And the way that we to try and age class female bats is very different and less precise, and also is bordered on being very subjective. So we eyeball basically a measure of traction alopecia, and traction alopecia is basically when it's a loss of fur around the nipple caused by nursing. So the fur around the nipples is lost because the babies are clamped on there, and the more times that she gives birth, the bolder her nipples are. So nursing midsummer, she's definitely at least 1 year old. But we can't really say whether a bat that has no evidence of nursing is definitely less than a year old, unless we look at other non-sexual assessment of age, which we get to in a little bit. But basically, we're kind of eyeballing it. It's very subjective, but we basically look at the level of traction alopecia to give us an idea of whether a bat is nursing for the first time, has nursed many times, or has not nursed at all. So occasionally bats cannot be obviously lactating. This is a little Coles pipistrelle here that you can just see a tiny little drop of milk. You can see if a bat is lactating by applying gentle pressure. It is and you don't really do that if you're unsure. If you think that a bat is what we call parous. Parous means that it's been nursing. Multiparous means that it's nursed many times. And nulliparous means that it's not nursed, or there's no evidence of nursing. So this is one of the things that we try to drive home as a matter of biological recording principles, is that we don't say this bat has never nursed. We say there is no evidence of this bat having nursed, because a female could nurse a bat for a week and then the baby bat dies, and so there's not enough time for traction alopecia to become really pronounced. But she still has given birth and she still has nursed, and so we would be making an assumption that was incorrect. So one of the things that we try to do is to not make assumptions and to only say what we can definitely say. And so we say nulliparous. Nulliparous means that there is no evidence of nursing. So here we go, a bit more into the terminology. A bat with traction alopecia is parous. A bat without is nulliparous. So sometimes you can see this picture here, there is no fur around this nipple at all. So she is multiparous. A lot of the time you can see a very old mother. Now bearing in mind that bats will live for many decades and give birth to one young each year. And so if they're a successful female, there will be a lot of evidence in old bats of traction alopecia, to the point where it looks pretty grim. So we can tell with some confidence that a multiparous female, but the younger the bat is, the less confident we are in being able to say that a bat she's given birth once or twice or whatever. So we don't record yearlings in females in the same way that we could do with males. Although there would be in some species up to a 30% margin of error of recording yearlings in males. So we basically just record mature and sexually mature. But in the case of females, it's basically they're sexually mature or there's no evidence. And then we would look for other things like dentition and calcification of knuckles, which we will cover. Here are a few example photos. So nulliparous. I think the thing that people struggle the most with is nulliparous to parous, because you can see here in this parous nipple, it's a little bit more prominent, but in the field, in the dark, squinting at a bat with your head torch on, it can be difficult to make a call. And you can see the multiparous bat. It's quite obvious. The nipple is darkened, and there is quite an area of flattening and traction alopecia around it. But the nulliparous and the parous, there's not an awful lot of difference between the two. And I would suspect that this lactating one, the third one along, there's still quite a lot of fur around the actual nipple. So it's hard to say how, it's a subjective assessment is ultimately what I'm trying to say here. So pregnant bats, we can say that they're 10 months old. Lactating bats, we can say that they are a year old or more. Multiparous bats, we can say that they are at least 2 years old. But nulliparous bats, we can't really say for sure. Okay, Becky has just posted some stuff in the chat if you want to have a look at her way that she remembers it.
It's worth noting as well that it's like at the start of the year, it can be harder to tell because the fur does grow back a bit.
Yeah, so. Indeed. Yeah, I mean, and our trapping season is a lot shorter at the beginning of the year than it is at the end of the year because we're constrained by the cold and we can't always crack on in April because often we get these cold springs. Most of the bats that we catch in the late summer, but when it's difficult, we need to rely on non-sexual assessment of age. So to that end, we look at knuckle calcification. So basically, bats, as you I hopefully you know, are mammals, and when mammals grow, in their long bones, so their fingers, their legs, their femurs, their tibia and fibula, and all that, the growth takes place at the end of the bone, and as a result, the end of the bone is quite soft, relatively, until they have finished growing, at which point the ends of the long bones calcify. And you can see evidence of this in the knuckle joints. So basically, if I'm hoping I've got some decent transparency photos in this. I've forgotten what I put in the talk because I wrote it like a month ago. But basically, as the knuckles calcify, they go from being transparent to being opaque, and they go from being long and sausage shaped like it is on this on the left hand knuckle to being round and sort of knobbly. The last digit for this to happen to is the finger bone of the proximal finger, the fifth finger. So it's its pinky. So remember, Jazz hand's facing towards you. It's its pinky finger, so it's the one closest to its body. That's the last one. And there's a picture which when we were doing that pop quiz a few weeks ago, there's a picture of a noctule that you can only see this in the pinky finger. So I thought I put it in here, but I guess I might not have. So to look for knuckle calcification, you will either do this over we have a little table lamp, like a desk lamp thing, or desk lamp. It's like a little bowl, a little glowing bowl that sits on the processing table, and we hold it over and look for the transparency in the knuckles. Or we use each other's head torches, which can be a bit of a pain because you're staring at someone's light and they can blind you. But we tend to lean more towards using these little camping lanterns now. Excuse the mites on the wing of this bat. So you can see the end of the finger bone here. This is the juvenile. You can see that transparent little window there. Oh yeah, there's the mite. Yeah. So the little transparent window on that knuckle, and the fact that that knuckle is sort of like long and sausage shaped and is not a tiny little ball, that's what you're looking for. You're looking for transparency. So next we do have that picture of that noctule. So if you look at the pictures of the finger bones of this noctule, the knuckle bones are round except for the knuckle bone of the pinky finger of that proximal digit. So that's sausage shaped still. If you look at that compared to the third finger, or the fourth finger, which are round and knobbly. So that's the way that it happens. Happens thumb, index, we go all the way down. So if you're not sure, make sure that you look at that and also check both wings. So just like when I said to you last month about checking wing venation, always check both wings. Same thing with this, always check both wings. So we can tell that this is a juvenile female noctule just from looking at the picture. All right, another horrendously subjective thing is dental wear. Dental wear is only very useful for identifying a very old bat. So you can see these a very young bat at the top and a very old bat at the bottom, and every other bat in between will be in the middle. And so if you see a lot of dental wear, you can say that it's quite an old bat. But again, there's no way to quantify that. You couldn't say whether it was 10 years old or 20 or 30. It is subjective but useful. So, for example, if you had, if you had a bat with unsure traction alopecia, you check the knuckles, you check the dentition, just to give you a bit more confidence in your assessment of the age of the bat. Right, another thing here, these are terrible photos, is the idea of the chin spot. So this is something that we're going to try and have a look at as we go through this year, but in Daubenton's bats, and I think pond bats, don't they as well, Scott? Chin spots. But you can, if you have a look on the bat on the bottom down here, there's a little dark patch on his chin, and these fade as they get older. So it's another subjective clue in our assessment. So this is the same species, but obviously no chin spot on the top one, but a little bit of a chin spot on the bottom. I will try and get some better photos this year. So health assessment. So we don't quantify the health of bats, but we do generally make notes if they are covered in mites, if they are skinny. If a bat was very, very skinny and looked like it was in a lot of trouble, we would take it into care, but that's never happened, and hopefully never will. Generally speaking, ectoparasites are something that the bats will groom off. A healthy bat will groom ectoparasites. So an ectoparasites basically will reach their peak during the maternity season, because bats don't switch roosts as much during maternity season, and so they can establish themselves. And a lot of the life cycle of some of these ectoparasites revolves around the ectoparasites being on the bottom of the roost and the young falling down and climbing back up to the mothers with a parasite load. But it is thought that one of the things that roost switching does is to keep ectoparasite loads down. So there's a few lovely photos of critters that we have found on bats. So ectoparasites are species specific. So you'll get a species of tick or mite, and that will only be on the species of bat that you found. There are some, I can upload some ID resources if it's something that you're interested in. So we have, this is a Cymex, which is a bat fly. No, bat bug, isn't it? Yeah, it's a bat bug. So there are lots of different kinds, and they're all species specific. So fleas, mites, bat flies, ticks, and bugs as well. So this one, Cymex, is a bat bug. And generally speaking, we just record what there is. And there are times when we've taken samples just to take some photos under the microscope, etc. But one of the things that we do that gives us an idea of how healthy a bat is its body condition index. So hold on 2 seconds. Right, this is clearly, I forgot to change the text on this, but damage can happen. Ignore the text on this, but damage can happen to wings. And we have seen bats readily flying with half a wing and just doing just fine. So generally speaking, if they're flying around and they're doing okay, then this is, it's nothing to be worried about. But then other times we get like bats in some of the bat boxes in the bat box schemes, you'll sometimes see them with these weird little lesions on them. We don't entirely know what's going on there, but and the damage to their wings can look quite horrific, but provided that there's not an awful lot of long bone damage, it does heal. We've seen bats basically with a tear from their wrist all the way down, splitting their wing apart, and over time it heals and it causes scar tissue. But the patagium is full of blood vessels, and it's a live tissue, and it's pliable, and so it will heal. So it's it's a torn wing is not a death sentence to a bat. Right, now this is where the actual this gets pretty gross. So they do get sort of infections, and a lot of bats have got a lot of sort of like, this is a noctule. They have a lot of glands on their face that can become clogged, just like any mammal can get sort of infections in their skin and stuff. And on the bottom right here, we're not quite sure what's going on here, but there's some kind of little growth going on the tip of his ear.
Yeah, that's a tumor.
Yeah, it's just pretty gross. Yup. So generally speaking, the body condition index, which I don't know if I do have a slide that goes into this, but basically the one of the ways that we look at weight for bats, because when we take the weight of a bat, that is because they vary. So the size of the bat varies, and a good measure of that is their forearm. So with any particular species, their forearm might vary by up to 5 millimeters, which doesn't sound like a lot to you or I, but if your forearm is only 30 millimeters, and then you have a bat of the same species that's 5 millimeters longer, then you would not expect those bats to be the same weight. And so we use body condition index, which it's a contentious idea, body condition index, but in general, it sort of smooths your data out for big bats and little bats within the same species. And so it gives you an idea of your population and individual bats, how healthy they are for their weight. So, for example, a few years ago, we had a brown long-eared bat. The only time this has ever happened, we had a brown long-eared bat diet processing. And so we wrote to Natural England immediately afterwards and everything. But basically, because we had taken the measurements of that bat, we knew that bat's forearm, that was for its species, it was, it's very sad. Scott has drawn a sad face. Great. Because for that species, it was a brown long-eared bat, had a 42 millimeter forearm, which is about as long as they get, and it was under 7 grams, which is about as light as they get. So being a very big bat for its species, having the lower end of the weight that a bat with a much smaller forearm just told us immediately that it was already in very poor condition. Also, it was covered in mites. So what we think had probably happened was that this bat was not well, not looking after itself, extremely underweight, and then just happened to go into our net. So it's one of the ways the body condition index is a really good indicator of how an individual bat is doing. We don't calculate body condition index on the night. We're much more concerned with like treat them and street them, just get the data we need and get the bats out into the wild where they belong. So this is something that we do after the fact. So we do record the parasite load, we record injuries, if they're particularly lethargic. I don't have a good photo of a skinny bat. A lot of times I used to have a lot of photos from doing bat care, but basically it's sort of like on there. I show you next slide. So basically just inside of the leg on the bum is where they tend to lose their weight. And so you will, I don't know, Scott, can you kind of illustrate on this bat where they'd be nipped in, where they'd be skinny? Yeah, so like, especially the bottom end behind the leg, they kind of lose a lot of their fat reserves there. And so when you get an underweight bat, they tend to have a skinny back. So other weird stuff that we record when we come across it is different pigmentations. And so we have a few bats at Merrian's Wood in the bat box scheme that have this pigmentation. And what's interesting is it helps us to uniquely identify the bat. So this is Eric. This is a bat that we've recorded a few times now. And there is another female bat which has similar pigmentation as well. Occasionally you get weird stuff like this noctule that we had that we could appear to only be able to move one side of its face. Very odd. But it's nature, wild, weird, and wonderful stuff happens. So a bit more detail on body condition index. So we take the weight, which we measure depending on the bat. If it's a small bat, we use a CD case. And if it's a larger bat, like a noctule, we use a cloth bag. And we tear off the weight of the cloth bag or the CD case. So you basically weigh the bat with there are two ways of doing it. One is that you attach the weighing bag to your scale and then you zero your scale. That's called tearing. Or you can weigh the bat with the bag, then weigh the bag and then subtract the weight of the bag, and then you know the weight of the bat. Either way, we take an accurate weight of the bat and then an accurate forearm length. So there's a couple of different ways we can do forearm. If we're just eyeballing it, generally we'll use like a wing rule like this, which is quite an easy way. What we like you to do is to use vernier calipers, which are a lot more precise, and they take a bit of learning how to read them. So yeah, we record all of these things. So, right. So now we are before I go into sort of the biological recording stuff, are there any questions about the first part? We've not got much longer to go. Nope. All right. Okay. We crack on. So a biological record is made up of 4 key components plus
Hold
a
on,
lot of.
Morgan. Got a question.
Yep.
Is there a high parasite burden? If there is a high parasite burden, do you treat them? No.
No.
Is the answer.
No is the short answer. If I got a bat in that was skinny, covered in parasites, I would consider taking it into care and then feeding up for a couple of days and then releasing, depending on the time of year. And when we have had bats in care, I mean, neither Scott nor I have done a bat care for a long time now because we're concentrating on survey work. But you can remove parasites from a bat using like a cotton swab dipped in alcohol, and that you can get rid of a lot of them. So I think if it if it was an inappropriate time of year to take a bat into care, but we had alcohol and swabs, I would consider giving it a bit of a swab down if it was really bad. But at the end of the day, you don't know what interference that you do is going to. You don't know what the knock-on effects of that interference is going to have on populations of bats. This is one of the arguments that people have about using antibiotics to treat bats in care, and that basically, if you are remedying something that happens to a wild bat and putting that bat back into the wild population, then perhaps the thing that should have been selected out that made that bat weak will then get passed on to its offspring. And then the offspring will have that same weakness. And so I think that it's a contentious issue in general. I think that treating and releasing wild animals is a contentious issue if you think too much about it, because the idea being that everything that dies in nature that's not anthropogenically caused is it should be happening. Animals die because they're not perfect as well adapted to that environment as other animals in that environment, and so that's how we adapt and how we become as animals. That's how we become better at living, to reproduce, to pass on our genes to the next generation. And so, how much are you messing with that by treating and releasing bats or other animals? We can say that about ourselves, though. Medicine is us sidestepping evolution, isn't it? I can see we have more chats. Is that another question that popped up? Because I can't see the chat, remember? Maybe I can't see the chat. Wait.
It's just me. I was just saying that the bat was already out there. It's living fine before we saw it and caught it in the trap.
Yeah.
We can't play God, as cool as that may sound. We would never have caught it and taken it off, so we can't really interfere with, as you say, the life cycle.
Yeah.
End of the day, it's natural selection. If that bat is going to die due to its mite load, it's going to happen. So It's just one of those. We kind of just leave it.
It's worth noting as well. It's like it's not the mite load that kills the bat. It's very rare that parasites will actively kill their host. It's they have the mites because there's something else wrong with them that might not even be treatable. So just because a bat's got high parasite load doesn't necessarily mean that by taking it into care and then removing that high parasite load doesn't mean that you're actually going to help it, because it could have something else wrong with it. And also juvenile bats, just because the way that these parasites, just the way the parasite's reproductive cycle works, juvenile bats are absolutely covered in parasites sometimes. So it's you very quickly get a stark contrast for what is a lot of parasites and a normal amount of parasites. What is a lot of parasites for an adult bat isn't a lot of parasites for a juvenile, and so on.
Yeah, absolutely. 100%. Okay, anything else on health, parasites, bats, biology? All right, we'll crack on. Biological recording. So to create a biological record, you require a minimum of 4 pieces of information. You need who, what, where, when. You need who you are, who the person identifying the bat is. That's the recorder. What, which is what species of animal or plant you have recorded, where you've recorded it, hopefully to a great degree of precision, and when you've recorded it. Without all 4 of these pieces of information, biological record centers and any biological recorder will just chuck your record out. So these are 100%. You need to have all of these pieces of information. So we will talk a little bit more about the different roles in creating a biological record. But generally speaking, I've got recorder determiner here, and that's a really important distinction to make. So get it into your head that the recorder is a person who says, I think this is a pipistrelle. The determiner is a person who says, I agree with you, that is a pipistrelle. So your recorder is the person who first goes, this is a brown long-eared bat. The determiner is someone who agrees with the record, who determines that your identification is correct. We make this distinction because a lot of the time, especially if you're recording cryptic species, you want 2 people to have made that agreement based on their field skills, their experience, and their independent assessment of what species it is. And so, when it comes to doing things like, I think I mentioned last time when I'm recording whiskered and brants in our project, if we get a small myotis into processing, I will look at the bat, identify it, and then I will text the head of processing my identification, and then Scott does the same. And if they ever aren't the same species, if we disagree, then it doesn't get recorded to species level. So we do this without conferring with each other, so that we know that we've both made an independent observation of the species. Because, you'll hear me bleat on about this for the whole season. Accuracy is the most important thing. If we're not accurate, we have no right catching, disturbing these animals. So other things that can go into a biological record include the method of recording. Did you use a bat detector, a bottle trap, a quadrat, a camera trap? Any number of things, a mist net, a harp trap, is it a bat box check? A method by which you gathered your data. It can just be field record observation, which is one that we use an awful lot. So the status of the animal. So was it feeding, flying, hibernating, alive, dead? And what life stage was it in? An adult, a larva? Was it in flower? Was it, this applies to all different taxa. These are universal principles about biological recording, not just bats. And you may also have media, sound files, sonograms, photographs, DNA samples, all different kinds of other supporting information. Most of the time it's some kind of media. And then you'll also have extra bats do not flower. Well, you'll also have extra things like the weight, the size, the health, the parasite load, all those other things that we were talking about that you might record, including sort of the assessment of age class and breeding condition, etc. So why do we record? Why are we actually recording bats anyway, or any species for that matter? So the idea is that we are feeding into atlases, both local atlases and national atlases. Knowing what species we have and what their distribution and their abundance is like, and having a very good idea of that gives us a good baseline from which we can get an early warning of population change. If we didn't have things like baseline data. We wouldn't have things like endangered species, because we wouldn't know that a species was in decline. We wouldn't have red listed or amber listed birds, because unless we have the data of how they're doing now, we are not going to have anything to compare how they're doing in 10 years' time. So it is a way of getting a heads up when a species is threatened. So it informs. This is one of the most immediate ways that biological recording, even in the project, out the project, whatever taxonomic group, if you go out and you see a badger set, if you record that badger set and send it to EcoRecord or upload it to iRecord, then that record will go to the local record center. And then when a company or an individual wants to do a planning development in the area, that record will trigger surveys, and then the results of those surveys will trigger appropriate mitigation. And so one of the best ways that you can support and conserve local wildlife is to create and submit accurate, timely biological records. And basically that is the way that the planning system works. So we get a lot of people write into the BAT group. We've had 2 people write in this week saying this housing development wants to knock down this woodland or wants to build on this brownfield site, and we want to stop it. So we want to know that if there's bats there, and we have to kind of write back saying, well, bats being present in the woodland isn't going to stop the development from happening, because if it doesn't have any protection as a site designation or anything, then it's not going to be protected. And bats being there won't stop that. What would records of bats in a woodland, particularly would trigger surveys, and then the results of those surveys would mean that either the person who wants to develop the site, the brownfield, the woodland, whatever, would then have to put up loads of bat boxes or do loads of supervised tree felling, and that would either mean that those bats were appropriately mitigated for, or that the development would prove to be prohibitively expensive and would happen somewhere else or be altered some way which wouldn't impact the bats. And so without the presence of biological records in the database of the local record center, then those wouldn't get triggered. And it is it's a fact that bats are protected and their roosts are protected, but their commuting routes are not, and their foraging habitats are not. So that's another huge reason why we do this. So if you can make your data available to students, to conservation organizations, then that can be used for analysis, to analyze trends, to analyze movements. There's, the amount of data that we collect, there's so much in there that can be used to infer information about change, distribution, conservation status, etc. And on a larger scale, climate change ecology is largely based on the analysis of biological records. So, for example, to step away from bats for a minute, there is most of you might have seen tree bumblebees, Bombus hypnorum. So they were new to the UK in I think 2003, 2002, something like that, and they're not an invasive species. They've not been brought in on crates or anything, but they've just made their way here through global warming, through climate change. We recorded the first one in the West Midlands in 2011, and now they're just bloody everywhere. And so the presence of that species and as the records of that species become more apparent, and those records, the distribution shown by those records becomes more northerly, that's evidence of global warming, of climate change. And so on a really large scale, on lots of taxonomic groups, that's what we can infer from biological records. And it's happening with bats. We see changes in things like horseshoe bats and various other species. We see we're getting a lot more records of vagrants.
A lot of bat workers down south point blank claim their bats do not hibernate.
Yeah, it's there's a lot that you can infer from biological records, and that kind of thing. What about not hibernating? That's not just information that you get from the who, what, where, when. That's when you first start to see the the value in recording whether species were torpid or not when you're doing hibernation surveys. If we get, like the battery habitat key database always records whether the bats were awake or asleep. It's just extra information. And in general, we want to record as much information as we can, given the resources available to us, the time available to us, and the constraints of disturbance on the species. So everything is always about balancing and justifying the disturbance that you're causing with the data that you're gathering. And that's the trade-off about why that's the whole point of the licensing system. So the idea of you having a bat license is because you are deemed capable of making a decision at whatever level of disturbance that you can do that disturbance without causing unwarranted deleterious effects on the species, its distribution, its ability to do its thing, and that any disturbance that you do cause is for the purpose of conserving that species, or is justifiable because of the data that you're collecting and the value of the data to the species and the favorable conservation status at large. Okay, so other things like I mentioned this before: red lists, amber lists, conservation ecology. So seeing trends in abundance and distribution give us red flags about when species are doing badly, and also when species are doing well. Things like red kites, which are just like pigeons down in like Milton Keynes and stuff now, were at one point very rare, and so it can give us the presence of biological records can give us an indication of the success of conservation efforts. And then also conservation genetics. So we're carrying out surveys. We're taking DNA samples, and so all those DNA, all of the data that we collect is going to be open source and uploaded to Biobank and available for people to do conservation genetic studies. And another thing, and this can be a bit woolly, is that to highlight biodiversity hotspots, both in our county, in our country, on the continent. But this is hugely affected by recorder bias. So the advantages, you can get information about species on a very, very large spatial extent. And you can also get very, very fine spatial precision depending on how good your records are. So if your records of all the species that you're looking at are recorded to 1 kilometer square value, that doesn't give you anywhere near as good data as if they're recorded to 10 meter precision. So we always try to record to a 10-figure grid reference whenever possible. And with, say, the Batless project, we're saying 8-figure grid references, so that we get precise information about what parts of are we recording bats in a woodland block only in the ancient woodland part, or only along the glades and rides, or is it, if everything is just recorded to that site, then you don't get very much information. So there was a study done in was it Scotland? I think it was near Glasgow, of woodlands of soprano pipistrelles. And because of the high resolution of the data that they recorded, of what species they recorded and where, they know that during the breeding season, during the maternity season, that the male soprano pipistrelles were pushed out to sort of fringe and edge habitats, while the females dominated the high quality foraging habitats. So the better your resolution, the more data that you can infer from it. So you get large scale, long term data, things like how are blue whales doing, globally. And most biological records are free to academia and conservation. So generally speaking, if someone has to, if a developer or a consultant on behalf of a developer approaches a local record center and asks for records of a certain, of protected species near a site, there'll be a charge for that. And depending on what county you're in, there will be different arrangements for that. So, for example, we collect a lot of bat records and all the bat records that we have go into the county database, which I manage. And because we have a service level agreement with EcoRecord, our local record center, we give them our records at the end of every year, all of our records. And this is different from how we used to do it. So basically, we have an agreement where they pay us a certain amount of money for our records every year. What we used to do was consultancies used to have to come to us, and we would provide them with a report. And because that was so lengthy and imprecise and being charged and charging them piecemeal for it. We just give everything to EcoRecord now. But anyway, so if you're a student or for conservation purposes or for research purposes, most local record centers will just give you data for free, the data that you're looking for, as long as you can justify it. And obviously you're creating work for people by requesting it. And there are some online sources for biological records as well. So where biological records come from ecologists, professional ecologists, amateur naturalists, students, park rangers, all different kinds of people working in the natural environment, either professionally or for hobbies, create biological records. Conservation groups, which don't always share their data, because a lot of the times people are working with protected species or species that are subject to persecution, like badgers. And so generally speaking, badger groups won't give you their data. And a lot of times badger groups don't give their data to local record centers. So, which, I mean, and we've, in our experience, we know of people who know where there are bat roosts of very important, very rare species, who don't give their records to the local record centers, respective of where they are, because they think that will mean that the location of these roosts will be in the public domain, and because they're sensitive, that those bats are a threat, rather than seeing the local record centers as a tool to trigger appropriate surveys and mitigation in the vicinity of those roosts. And if nobody knows where they are, they're just going to fall foul of holes in the planning system. So universities create them, and I've been saying this for years, that the amount of students doing, even just at my university, the number of students doing local surveys and just the biological records that must be in the backlog of dissertations at the University of Wolverhampton of local records of different species must be astronomical, and they're not generally speaking not submitted to local record center. So these slides are adapted from a lecture I do for the MSc students at the uni, and so I really push them all to kind of like send their local records in, but yeah, universities create an awful lot of data that doesn't always end up in the record system. Museums have vast collections, especially entomological collections, that have never made it into the record system. My MSc thesis was looking at collating museum specimens, records of particular species of bees, and doing analysis based on those. And there are county recorders, and one would hope that county recorders they usually get a county botanist, county bird recorder, county bat recorder, and then usually like ARG groups and stuff like that. A lot of times their data doesn't make it to the local record centers, and it needs to be in the local record centers for it to actually have any beneficial effect on the planning system. And generally speaking, recording schemes. So this will be stuff like the Big Garden Birdwatch, and the BCT National Bat Monitoring Program. And that stuff does come down to, like, I get sent the results of the National Bat Monitoring Program, and that comes to our database, and then we send that to EcoRecord every year. So usually national recording schemes are pretty good about stuff like that. So I wanted to talk a little bit about atlases, especially because we have the new Batlas project relaunching, I should say, this year. So I'd like to ask you, do you think, and you put it in the text, yes or no, do you think that this is a good definition of what an atlas is? Do it. Type it. Do it. Nobody's playing. Nobody wants to guess? Someone be brave. Come on. Is it a good definition? Atlases are geographical representations of the distribution of species. Oh, you chickens. You big chickens.
I will go for you. I'm going to say no.
You're going to say no, okay? And Elizabeth said yes. Any other advances? Becky says yes. Okay, could be more specific. You got 10 seconds, guys. Come on, be brave. I'm sorry, my phone is making noises. Here we go. So agree with Simeon, and yes, generally speaking. All right. So, it is what they are supposed to be. is the answer to this? It is what they're supposed to be. Generally speaking, that should be the definition. So we'll take another bit of a look at it. So, an atlas is a map of distribution of the records of species, not the distribution of the species. So that's where when you look at an atlas, it is always biased. Things like favored good sites, sites near recorder homes. So hopefully I have put in a few examples of this. So here you go. You've got distributions of frogs and toads here. So the reason that I have put this in is because if you look, can you see where Birmingham City Centre is on the Common Frog? Right, so that is Birmingham City Centre there. You can see that a lot of recording happens here. That basically you could be forgiven for thinking that frogs and toads are both almost absent from Sandwell and Wolverhampton, and we know that that's not true. Another one. This is from the Mammal Atlas 2003. This is the distribution of water shrew and the distribution of house mice. And so you could be forgiven for thinking that there are twice as many water shrews in the county as there are house mice. And quite clearly, that's absolutely not the case. Yeah, Becky has just put, it's often said that an atlas is a distribution of map of recorder effort, and it really, really is. There's no way there are twice as many water shrews as house mice. What it is that when people see a water shrew, they will record it. If people see a house mouse, they don't record it. So that's an example of one type of bias. You have taxon bias. So you're either recording the things that you're interested in, because like I record, obviously I record a lot of bats. Whenever I record amphibians and reptiles, I give my records to Scott. Usually we record them together anyway, and then he submits them. And if I record anything else, like mammals, whatever, I put those on iRecord, generally speaking. And that makes its way through a verification process and then to the local record centers. But I don't tend to record plants, and I see hundreds and hundreds of them. And I don't tend to record birds either, even though I'm a birder and I go out and I see birds. Unless I see something particularly rare, it's like I'm not going to record every blue tit that I see. It's like Lee and I went out yesterday for a walk and we saw a tree creeper and I didn't even record that. So, so Becky's put, we know rats are pretty much everywhere, but do you record this? Most people wouldn't, yeah. So we don't record common species. We record species that we think are charismatic or that are particularly within our area of interest. So where does bias happen? So bias can happen in species abundance. So for example, if you are, all of these things, bias can happen with species abundance. And when you look at abundance on a map, like the number of records on a map, and we're going to look at how different maps can display different information. But basically, species abundance can just be because they get more often recorded at a site that's easy to access. Or a site that more people go to. So like, for example, Sutton Park is one of our most recorded sites in the county, because a lot of people go there to record it because they know it's a good site and they're going to see cool stuff. There is a local conservation, there's a local Sutton Park Natural History Society, and there's a lot of things at play that cause people to record more at Sutton Park than they do at, say, Barbeacon, which is a mile away. So bias happens in lack of conservation value. So people don't see brownfield sites or urban woodlands as having any value, and so they don't get recorded, which is why every new site that we go to do bat trapping, we record new species for the site. At most, there will be a pipistrelle record for it, but that's it, because no one has done any recording there. No one has done any distinct survey effort there, because there has been this assumption that they would just have common or ubiquitous species. Ease of identification. So, if an animal is easy to identify, they're easy to be confident and make a biological record. If you're not sure, then you're less likely to record something, which is a good thing, because it's better that people only record stuff they're sure about than make guesses and then have erroneous records submitted. Things like plant height. So this is an actual thing. So you are much more likely to record species that live, that spend their time on at sort of like knee to waist height vegetation than species that live on the undersides of leaves or are in the lower vegetation or in the tops of trees. So that's in a sense an access thing, but it's also an effort thing. So if you're walking through a nature reserve, even doing something specific like a BioBlitz, generally speaking, you're going to record what is easy to see. And one of the other things that makes it, that exacerbates this effect, is if a species is brightly colored. So you're much more likely to have your eye caught by something and record species that are brightly colored than species that are not brightly colored. So you're much more likely to record a bullfinch than a tree creeper in a woodland, because bullfinches are bright and tree creepers are brown. So and also you would be going to favorite sites. So I'm sure that you all have your favorite sites to walk around. And you might walk through a park to get to a woodland and record stuff in the woodland, but you wouldn't bother recording stuff that you saw in the park on the way. Stuff like that. And then you'd be recording in your taxonomic area of interest. Like I said, I don't record birds. So, because I basically record mammals and herps, and that's pretty much all I record. And I do record some inverts, but I used to be mostly inverts, really, when I was doing my masters and stuff. So, and that will change throughout the years. But also then the skills of your recorder. So if you're obviously going to be learning to identify cryptic species of bats in the hand, at the moment you may not be able to do that, but in a year or two's time you will be able to do that. And so you're much more likely to be recording, more difficult species. And all of this causes uneven sampling over space and time, and uneven sampling over survey effort. So there are so many things at play making the atlases wrong. So things that they show us. So these are some examples. I'm going to have a look at so basically you can plot biological records change over time. So these are all from the Southeast Mammal Atlas. I think that they are mostly whiskered bats, I believe. I think they're all whiskered bats. But basically, you can show species loss. You can show the range of species. Now, this is the range within these few counties, bear in mind. And then you can show distribution. And so you've got sort of the Isle of Wight down there. I think it's the Isle of Wight, isn't it? And then you can see there's a real big hotspot in the middle. Now, we don't know whether that's because there's a big habitat change. Is it more wooded in that bit? I mean, that's where aerial photography and a bit of other data to help you interpret would help. But basically, the distribution, remember, it's distribution of records, not distribution of the species. The only way that you would be able to accurately plot the distribution of the species over your study area is to conduct the same amount of exactly the same survey effort in every square, and then present your data, which is what the BATLAS is about. So the reason that we have all of this information about bats over the decades for Birmingham and the Black Country, but we don't know how much of that is biased. And so by doing a standardized survey with a clean slate, and doing the same amount of survey effort in every 5 kilometer square and every 2 kilometer square, we'll be able to say, this is the distribution of our bats. And if you ever see sort of like an atlas that will say provisional atlas, a provisional atlas is an atlas of all the records that there are. So our provisional atlas that was published in 2017, that was all the data up to 2014. That was provisional because it was not based on a standardized survey. It was based on all the records that we have. Now, when we do produce a full atlas, we're going to do a 5 kilometer one next year, and then 2 years later a 2 kilometer one. We'll be able to compare that with the provisional map to say how much have we filled in, how much more do we know about bats. But it gives us a good baseline. So you can look at recording effort, you can look at range, and you can look at species richness. So this is a pretty cool one. So the hotter the color, the more species are recorded in that tetrad. On this map, which is pretty cool. Again, incredibly biased, but that's the nature of it. And then I've had conversations with people before where they've said that. So because I do a lot of recording in Warsaw, I'm skewing the maps, and that's impossible to do. The maps are already skewed. Every record that you create, no matter where they are, is a step towards accuracy if you're recording new species. It's just that unless we can disperse that effort over an even area, we'll never get an accurate picture. And that's the whole point of doing the Batless project. So when a record gets submitted, so I'm an iRecord verifier for Worcestershire, Warwickshire, Staffordshire. And when we get records in, we either accept it, say that it's plausible, reject it, or query it and ask for more information. And I thought I'd just throw in a few examples of stuff that I've had pretty recently. So lack of independent identification is a huge red flag. So a reliance on auto ID for inexperienced people to submit records based on just because their EM Touch told them it was, and lack of adherence to process and this whole idea of, like I said to you before, about making assumptions, which we don't do. We record to the highest level of detail of which we are sure. And also things like I said, anatomy, not pelage. And how likely is it that species is there? And this is one that I got very excited about when I first saw it. As you can imagine, it's a record for within our county boundary of a barbastelle, which would make me do a cartwheel. However, the recorded comment was found using a bat detector. Yeah, Becky says when she sneezes, her EM Touch thinks she's a nocturnal. So yeah, this was a huge red flag. But having said that, if we're going to get barbastelle, this is the part of the county we're probably going to get it, the western edge. And this is out by Whittick Manor. And so maybe it was, but it's certainly not likely. And the method makes me think that it's unlikely because it was found using a bat detector. So I emailed this person to check the record, and it was an EM Touch. And so I've chucked the record out, unfortunately. Okay, so here's another one. Going on jizz. So this is really cool. I found this out recently. The term jizz was published by T.A. Coward in 1921, who heard it from an unidentified Irishman. So the idea of jizz is that some people believe it just is. So it's a birding term, generally speaking. You'll just go, oh, based on that bird's jizz, I think it's a magpie or whatever. Just the way that it's acting. But other people think it is a military term, general impression, size and shape, jizz. Either way, it's not science. Jizz doesn't fly when it comes to biological records. You need evidence. You need to have some kind of evidence, either the evidence being your field observation based on your experience and your knowledge and skills, or some kind of photo evidence, DNA, all the things. Okay, so basically, if you have a look at this one, record of Serotine. So the bat flew around Halfords for about 90 minutes in well light parts of the warehouse. So I was able to get a good look at it and was able to see it had a very pale belly. It was also seen by many people before it left safely. I have seen large bats in this area and always suspect them to be Serotine, but unable to make a positive on ID. So this person recorded what they were looking for. Right, so this basically, they didn't use a bat detector. They saw a large bat flying around a warehouse and decided, based on the fact that they thought its belly was pale, that it was a Serotine. But also, clearly biased because they already thought that there were serotina in that area, but had never confirmed it. And so I took this one out as well. And lack of verification data. This is quite spectacular. So record of Nethusius pipistrelle. So in August, and their recording method was visual sighting from a distance. Now, I wouldn't record an Enthusiast pipistrelle based on a visual sighting from a distance myself, and I've been doing bat work for over 20 years. So this person is either, the other thing is it's a BCT sunset sunrise, so it's possible that this is a known roost. So what I did with this one was I emailed them and I'm like, is this a known roost? Because it might be that they've gone, visual sighting from a distance, because they just go outside, they do the BCT sunset sunrise, and it's just a count of a known roost. They count the bats coming out. But there's no evidence in this submitted record of their method of identification. So this one was queried, not rejected, but queried. So the idea of a lack of verification data is an example of that. So all this comes down to, are we gathering the data that we need? Are we prioritizing welfare? Your welfare first, remember from your induction, human welfare, bat welfare, data. And are we then prioritizing the data? So we want species, sex, breeding condition, DNA. Then if the bat is continuing to not be stressed and is okay, then we'll do things like biometrics. So at every stage of recording bats, as we go through our project, we are assessing the stress levels of that animal, and every single step of the way, you're making that judgment as to whether is it worth the data, the stress that we're causing? Because of course, anytime you handle a wild animal, it is under some stress. So are we adhering to the bounds of our license is a huge one. And I've seen people breach their project licenses in the past, and we are super strict about ours. If you find that you're in a project and you need to be able to do an extra technique, you just get it added to your license. Just do it properly. Just talk to Natural England. They're amazing people. And this is the big one, are we refraining from conjecture or guesswork? So we record only what we're sure about. We don't make inferences about species, sex, breeding condition. We don't make inferences about any of that. We record everything. We record what we're sure about in as much detail as we're able to record, using the time and resources available to us, balancing the needs and welfare of the animal. So that is the kind of like the package of our whole approach to recording. And this goes for, obviously, all of this stuff goes for bat box checks, as well as for trapping. So we record the site and the grid reference. So we have a grid references for each of our nets, so that we get that fine resolution data. So we record the trap and the net that it was caught in, or the bat box that we pulled it out of. The date and the time of capture. So these are things are the scribe. The scribe is the person who makes the notes. The scribe is the person who uses EpiCollect 5 and records the data that is told to them. Now, the reason that we record the scribe and we record the handler, which is not necessarily the recorder, because it might be that Scott brings a bat from the net and says, it's a brown long-eared. Scott's a recorder. And then Becky takes it out and starts processing. She's the handler. Tasha is writing the notes. She's the scribe. So and we record as you go through your EpiCollect, whether you're doing bat boxes or whatever, and a lot of times during multipass, we record who the scribe is as well, because I need to know if a mistake is made, who's made it, not so I can tell you off, but so that if you're getting something wrong because I haven't trained you properly, or because it's something that you don't know, then I can address that and it doesn't happen again. So yes, right, yeah, Becky has put, in that case I would be handler and determiner, as I would agree with Scott. I know, don't just agree with Scott on principle, Becky. You could we've had that happen before, where someone who was quite experienced came to a trapping survey and identified a bat as a whiskered bat, and then Scott assumed that that person had done the identification correctly, And it turned out that when he came back, he goes, "That was a funny looking whiskered bat to me," and I went, "That was a natterers." And it was literally, it's very, very easy to respect someone, to maybe feel out of your depth, to have a bit of imposter syndrome, and as a result, to not question everything. You should question everything. You should question me. Everybody makes mistakes. Everybody. So scribe handler is a person who handles it, and the recorder and determiner, as we've said, is the person who records it, and then the person who question everything, Tash. And then the determiner is the person who says, yes, I agree, or no, I don't agree. And then we go through a period of having to get more details or evidence. And then we record the species. So as you can see, up to that point, we have done who, what, where, when. But then we record sex, breeding condition, DNA and volatiles, morphometrics, biometrics, and then any notes and observations and photographs. So this is the order in which we record things. And in the first lot, basically the bat is got out of the net or the box and is put into a bag and then is chill, ideally, unless it's a nocturnal because they just lose their shit. Then they're chill in the bag. When it becomes a matter of being efficient with your handling and your ID and trying to minimize the time that you've got a bat in care, a bat in the hand, is the point when you take it out of the bag and you go, what species is this? What sex is it? What's the breeding condition? And we do all of this as quickly as we can, but without compromising accuracy. But in general, we record who is making the mistakes and where, so that we can address your training needs. We are nearly at the end. So how long do we gather data? We gather it until we have what we need, or until the bat is stressed, or until we cannot reasonably justify further disturbance. So that is something that we have to keep in our mind the whole time for whatever type of survey that you're doing. Even if, hopefully next year we'll be allowed to do internal inspections again and hibernation surveys and stuff, and that applies for that. Anytime you're doing anything which is a licensable activity, which has a potential to cause disturbance, and that includes doing things like using torches. All of this stuff is licensable. All of it has potential to cause disturbance, and at every single stage we have to constantly be asking ourselves if what we're doing is justifiable based on the data that we're gathering. And again, we gather as much as we reasonably can, accurately and ethically, in the time allowed, with the resources available. So that is the basic, the whole approach for the whole project to data gathering. And hopefully you will take this on and then you will apply these same principles once the Urban Bat project is long gone, that hopefully this gets drilled into you and you become better ecologists and better naturalists because of it. Okay, so buy this book, Dietz and Kiefer. But in the run up to, if I can recommend any book about bat ecology and anatomy and identification to you, it is this one. So I am gonna also share, bear with me one moment before we finish. Oh my God. Close this down. Just wanted to share with you some stuff about the Batless project while we are here. So you will have heard me talk about the Batless project. Right, you can find the Batless project website. It's on the Brumbats pages, if you've got brumbats.org.uk, or you can go directly batlessproject.co.uk, and it will bring you here. And here you can find a link to the existing provisional Batless, so the Batless of the non-standardized records up to and including April 2014, which is here. But what we're hoping to do is to update this with a systematic survey of all of the county. And so it happens in 2 stages. In stage 1, we're going to be looking at 5 kilometer square resolution. So there are 40 5 kilometer squares in our county. And we will be surveying each one of those by placing an audio moth static detector in it for 5 consecutive nights during the survey season, which is going to be mid-July to the end of September, 10 weeks. So each of the 10 members of our bat group committee is adopting 4 of these squares and taking on 4 volunteers, up to 4 volunteers. And basically it will be a matter of collecting the equipment, going out on site, choosing a place to put it that's nice and secure, and then leaving it out for 5 nights, and then going back and collecting it and then returning it to either myself or to Tasha. And this will happen if all being well, we'll get all 40 of these done this year. And at the end of this year, we'll publish an atlas at 5 kilometer resolution of the distribution of our bats. And then next year, every one of the sites that these were recorded this year will be attributed to their tetrad, and then we'll go on to start the batless at tetrad level, which is 202 tetrads. So we'll have 162 tetrads to do in the following 2 years. And the idea is that every one of the people who volunteered this year to help one of the team leaders to do their 4 quadrants will then adopt 1 or 2 tetrads for the following year or 2 years. And we're hoping then that within 3 years, we will have this even systematic baseline survey of the bats of the county. Fingers crossed. So you can get involved if you want to. It doesn't take an awful lot. As I said, it's a matter of going out, stick a bat sector on site and coming back and get it a week later and then returning it to one of us. So there's not an awful lot to do. And you'll find out the results of your survey before this time next year. So we're hoping sort of over Christmas. So there's not an awful lot to do to take part, but the idea is that you take part, you understand how the method works, and then you can adopt some tetrads of your own next year. So you can sign up to do it here. What you would do is you'd fill out this form after looking at this map. This map is updated constantly anytime someone adopts something. So say you live in Wolverhampton, right? So basically the grayed out ones are adopted. The non-grayed out ones are not adopted. So say you want to do this one here, SO89E, you would fill out your form here. And you write SO89E here, hit send, that sends me an email, and then I would update the map, and then you would be contacted at some point in the next month or so by Tasha, who is the team leader for that area. So that's how that works. So if you're interested in helping to create one of these, like a proper batless, not a provisional batless, then that's something that you can get involved in. We will be doing some testing. Like I said, the survey season for batless doesn't start until mid-July, but we will be out all spring doing field testing and making sure that the procedures and the equipment and the sound analysis all work well. So, as I said, batlessproject.co.uk, or you can find it through the Brown Bats page. The other thing, those of you, in case you missed it, is that that on the Urban Bat Project page, you can now click through and watch all of these training videos on there, and I will post the link to it again. So, any questions?
I noticed during the PowerPoints that you mentioned iRecord a lot. I've been using iNaturalist. Do you think one is better than the other in terms of recording species? I just never heard of it before today. So
yeah, iNaturalist is I use iRecord. iRecord has its systems in place where everything is sent to the verifiers, and then from the verifiers, once the records are verified, it's sent to the local record centre. So if you're someone who does a bit of recording in Staffordshire and a bit in Herefordshire and a bit in West Midlands, then iRecord is really good because you don't need to even keep your own, and you can download your own records as well. Shall I show you iRecord just in case people want to see it?
Yeah.
I think the problem with iNaturalist is they don't actually submit their records to the local record centre.
Yeah,
so it's better to use iRecord. But
the advantage with it is that it's international, so you can use iNaturalist if you're on holiday. I use it when I go to Florida. So, right, let us have a quick look at iRecord. So, so this is you wouldn't see this. This is I've landed on because this is my verification page. So for example, you can look at the records that I've accepted recently. There you go. Nice Lesser Horseshoe in Worcestershire. but she sent me a photo, which is pretty cool. So I know that she sent me a photo, so I'm just like, yep, no problem. Send the record to Cody as well. So it's so I just forward that. But basically, iRecord is like this. It's an app on your phone, and you basically just fill out what you've seen and where you've seen it. So you can enter them on here, add photos, put exactly where you are on the map, that kind of thing. But the app just does it all automatically. I highly recommend it, especially for if you are recording things outside of the county, because otherwise you have to do what Scott ends up doing at the end of every year, is splitting each of his records into a data set for each of the local recording schemes, the biological record centers, and has to send them all off in separate emails. But if you are recording bats and you're not with us, then if you go to the Brumbats website, you can submit records here. Oh, there's Andy. And then again, here we go. There's all the information: who, what, where, when. We're only interested in Birmingham Black Country Records, and then there's a link there to iRecord. And then there's a link to sort of finding your local biological record center if you're outside of the area. But basically, this is it: the who you are, what you've seen, where you've seen it, and when you saw it. And then this stuff, method, all the stuff that basically, and all that does is populate a spreadsheet. And at the end of the year, I drop that into MapMate, export it, and send it to EcoRecord, and then we get 500 quid. So yeah, I mean, submitting records, I don't care how you do it. Obviously, I prefer you did it through, I prefer your BAT records came in through this, because that helps support us in our service level agreement with the local record center. But it really doesn't matter, because at the end of the day, what matters is that the records get into the planning system. However, iNaturalist, they do lots of things, like they do like competitions and sort of like international. They did one that we took part in this year as part of one of our Bats Live walks. It was sort of like a competition to see who could record which city in the world could record the most records. And it's an international thing. So iNaturalist is a great thing to have on your phone. But if you are going to be regularly recording local stuff, then I would definitely use iRecord. There is also iSpot, which is a quite good, almost like a game, kind of, because you get points for things, and it's just but it's a good, like, dial-a-nerd. If you find something that you don't know what it is and you upload a picture of it, some nerd will identify it for you. So okay. iSpot is also useful, but for recording, iRecord is what I recommend.
Okay, brilliant, thank you.
Anybody else? Dun, dun, dun. Right, I'm just going to show you the iRecord app. Oh, it's not focusing. Focus. Focus. Oh, there you go. iRecord. It's got a little dragonfly on it. So the other thing that you'll need for this year is EpiCollect 5. So if you haven't downloaded EpiCollect 5 already, I would do that. Right, Scott, anything you want to add before we sign off?
No, that's it. Really.
Okay, so yeah, just any questions, just give us a shout. The next talk, well, in 2 weeks we have Sounds Analysis Sunday. I think that will be the last one. And then 2 weeks later we have got Bat Ecology. And then I think there's another Sounds Analysis Sunday. And then the final is going to be how Multipass works. And that will be the first one in April. And then hopefully by then we'll be out and surveying. Not long. Spring soon. Bat soon. All right. Okay. Well, thank you, everybody. I am going to stop recording and sign off. Any questions, just give us a shout on WhatsApp. Okay. I shall upload this as soon as it's done processing. Okay. See you guys.
Thanks, everyone.
Bye.